Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is meant for 14th October 2020. We are into the revision mode and we have been revising a very important chapter as far as your CBSE board exam is concerned as well as your career is concerned, that is molecular basis of inheritance. Again, I want to stress on this that give a lot of importance to this chapter as it contains maximum marks in your CBSE board exam. Plus, in future, you can always become a molecular biologist. Plus, you can be proud of working with Nobel laureates. That's the thing. Like I said, Professor Crick. Okay. So, moving ahead with that, transcription. We're going to talk about today transcription. What is that? It is the process of copying genetic information from one strand of DNA into RNA. This is important thing. So genetic information is copied. In transcription, only one segment of DNA and only one strand is copied in RNA. The adenosine forms base pair with uracil instead of thymine. Of course, we said long back that thymine will be replaced with uracil in the RNA. So transcription is a process of making copy of genetic information stored in DNA strand into complementary strand of RNA. That complementary strand of RNA we call it mRNA. So basically you must remember transcription means DNA becoming mRNA. This is the simple one. You must remember DNA becoming mRNA. So all the genetic information from DNA is copied into the complementary strand called mRNA. That's why we are intentionally calling it as mRNA, messenger RNA. All the genetic information in the form of messages will be coded in that. So transcription of DNA includes what is required for this. First and foremost, a promoter should be there. Okay, and the structural gene should be there. Second, and a terminator will be there. So these three are compulsory. Transcription of DNA includes promoter, structural gene and a terminator. The strands that has polarity 3 to 5 prime act as a template and called template strand, and the other strand is called coding strand. So this is how we are saying. You can see the blue line 3 prime to 5 prime, and you have got the 3 we said. The red zone one is the promoter. Then you can see the structural gene, and of course you have got the black one, terminator, the meaning terminates. So it is the 2 strands. One is called a template strand, another is called coding strand. So this is how the structure of transcription code will be there. So transcription requires an enzyme called Rna Polymerase. The entire or complete enzyme consists of a core enzyme, and of course, if you recollect from your memory, we said sigma factor. So sigma factor is a must. The sigma factor recognizes the start signal on DNA and directs the binding of the enzyme to the promoter region. This is the first step. So there has to be sigma factor, and it is thus enhances the specificity of transcription. Eukaryotes have 3 RNA polymerases. If you recollect from your memory, we talked about RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. RNA polymerase 1 is located in the nucleolus and transcribes for rRNAs, that is ribosomal RNAs. RNA polymerase 2 is localized to the nucleoplasm and is used in hnRNA and mRNA, and RNA polymerase 3 localized to the nucleus, possibly the nuclear plasm or nucleoplasm interface, and transcribes for tRNA, transfer RNA. So you got 5S rRNA and snRNAs. Prokaryotes have, on the other hand, only one RNA polymerase which synthesizes all types of RNAs. So RNA polymerase in E. coli has 5 polypeptide chains. Okay, this is the background that we talked. If you remember, So the segment of DNA that takes part in transcription is called transcription unit. The transcription unit. It has three components, like we said in the transcription unit. So this whole thing we call it as transcription unit. Transcription unit. And this transcription unit has three parts. What are those? First and foremost is the promoter, that I said red marked promoter, and then you got a structural gene, this whole structural gene, and then a terminator, the black one, this region, terminator. Besides a promoter, eukaryotes also require an enhancer. Promoter has different parts for attachment to various transcription factors. In many cases, the promoter has a AT-rich region, and I am saying this intentionally, that AT-rich region is called Tata box. So there will be what is called Tata box, TATA box, Tata box. Okay, and structural gene is component of that strand of DNA which has 3' to 5' polarity. So structural gene has got 3' to 5' polarity. This strand of DNA is called template strand, as I said just now, or master strand of antisense or negative strand, or we can say antisense strand. The other strand which has a polarity of 5' to 3', you are saying here 5' and 3', and this is called the 5', it is non-template, displaced during transcription. This non-template strand which does not take part in transcription is also called sense or coding strand. You see, coding strand is green one, sense or coding strand, or we say, there we said that it is negative, the template strand is a negative one, here it is the positive one. So this is just maintaining polarity. So because genetic code is present in the strand is similar to genetic code based on mRNA, except that uracil is replaced by thymine. A terminator factor called rho factor. When we come to the terminator factor, here we said sigma, and here it is rho factor. So how do you write this way? Rho factor is required for termination of the transcription. So post-transcriptional modification, transcription in eukaryotes occurs within the nucleus, and mRNA moves out of the nucleus into the cytoplasm for translation. The mRNA is proceeded or processed from primary RNA transcript, a process called maturation. Initially at the 5' end, a cap consisting of 7-methyl-guanosine or 7-mg and a tail by poly A at the third end are added. The cap is chemically modified molecule of GTP. What is GTP? Guanosine triphosphate. The primary mRNA transcript is longer and localized into the nucleus, where it is also called hnRNA. What is that hnRNA? Heterogeneous nuclear RNA, or it is also called pre-mRNA. The primary mRNAs are made up of two types of segments, non-coding introns and coding exons. So you got non-coding introns and coding exons. The introns are removed by a process called Rna splicing, and the enzyme ligase joins two adjacent exons. So this is what you need to know about this, and the difference between template strand, this is strand, and coding strand. It is DNA strand with the 3' to 5' polarity, and coding strand, of course, as you are seeing the green line, it is 5 to 3 polarity. Acts as a template for transcription and codes for RNA, and does not code for any region of RNA during transcription. So that's why we say nonsense and sense codon, positive and negative codons. Promoter is located at 5' end, and that bind the enzyme Rna Polymerase to start transcription. Just now I said sigma factor also helps in the initiation of transcription. The terminator is located at 3' end, of coding strand and usually defines the end of transcription, where Rho factor will bind to terminate transcription, end of the transcription. Now this is how we can draw the diagram. See here, RNA polymerase enzyme, and sigma factor is there, promoter region 5' to 3', and now there are 3 steps in the total transcription. First one is initiation. So initiation started. For initiation, what are the 2 things that are must? One is the RNA polymerase, second is the sigma factor. And then second one is elongation. As the RNA polymerase moves ahead, you can see the opening of that. And as it moves into the structural gene component, we can see the coding of RNA. The RNA, you can see the yellow portion is getting out. And then termination. Once the Rho factor, the end factor, yes, Rho factor and RNA polymerase. The Rho factor gives a signal for termination. Once the termination of the transcription takes place, RNA has come off. So exons are those sequences that appear in mature and processed RNA. Exons are interrupted by introns. Introns do not appear in mature and processed RNA. There are no introns. So in eukaryotes, there are 3 different RNA polymerase enzymes. Just now we discussed that. 1, 2, 3. They catalyze the synthesis of all types of RNA. And what are those RNAs that are synthesized? RNA polymerase 1, ribosomal RNA. RNA polymerase 2, messenger RNA. RNA polymerase 3, transport RNA. So the mRNA provides the template. tRNA brings the amino acids and read the genetic code, and rRNA plays structural and catalytic role. Now I want you to focus on this structural and catalytic role during translation. So what are the differences between DNA replication and RNA transcription? In DNA, 2 new molecules of double-stranded DNA are produced when DNA replicates. Only new molecule of single-strand RNA, that is mRNA, is produced. Adenine on one strand binds to thymine 80 on the new DNA strand being created. Adenine on DNA binds to uracil on the new RNA strand that's created. The entire chromosome is replicated. Only a small portion of DNA molecule is transcribed to RNA, and this varies based on the cell's need at a time. Enzymes, DNA polymerase is compulsory. Here, RNA polymerase, as we discussed now, occurs inside the nucleus, occurs in the nucleus, but once mRNA is formed, mRNA comes out of the nucleus into the cytoplasm. So the primary transcript contains both exons and introns and is non-functional. It undergoes the process of splicing, RNA splicing we said. The introns, removal of introns is splicing, right? So the splicing in which introns are removed and exons are joined in a definite order. Who is joining these exons? Ligase. An enzyme called ligase, Ligase. And the hnRNA, heterogeneous nuclear RNA, undergo additional processing called as capping and tailing. In capping, in unusual nucleotide, like we said, methyl guanosine triphosphate, at the 5' end of the hnRNA. In tailing, polyadenylate, okay, tail is added at 3rd' end in a template at the independent manner. So there is addition, okay, that is in RNA polymerase binding site. So you got 3. The polymerase continues transcribing unit. It reaches the termination site of the mRNA transcript and is released and is ready for translation. So next we will be studying about genetic code. What is the genetic code? Okay, and the characteristics of genetic code. And once we come to know that, we will be proceeding to what is called Ubel hypothesis. And then we go for translation, that is from mRNA how proteins are formed, which are the secrets of our body. And this we will be studying in our next class. Until then, keep practicing the structures, particularly DNA. Write each compound, particularly nitrogen bases, 5 nitrogen bases. You have to draw, understand the carbon arrangement, okay, in the chain, and then how to attach. The sugar molecule is to be attached one side to phosphate molecule, another side to nitrogenous base. So that is a nucleotide. That's why what is DNA? We say polynucleotidal chain. At least you write 2 nucleotides. Then you understand how DNA structure is formed. Then you draw transcription unit. So these things you must do it today only. Then we will move to translation in the next class.